Hello students, this is Shayista from Department of Studies in Microbiology, KSVU. Today's topic which I'll be dealing is the mechanism of enzyme regulation. So this topic I can say is the most tricky topic in the syllabus, not because it is difficult, but because many students don't know the basic terminology which is used. So to begin with, let me give you a basic of the enzymes. There are mainly three types of enzymes based on the nature of their synthesis. That is when they are synthesized, in which condition they are synthesized. On that basis, they are divided into three types. The first one is constitutive enzymes. These enzymes are present in the cell almost all the time in a particular given concentration. So you take a cell and they are present in it, irrespective of the composition of the medium in which the cells are. They are very vital for the cell functioning, and you can tell that these are the enzymes which don't get regulated easily by any substance. That means their regulation is very specific. So the best example which I can give here is the enzymes which participate in glycolysis and Krebs cycle. As students, you all must know that Krebs cycle or the glycolysis are the universal cycles which are highly conserved. That means they don't change. You take it from the E. coli to the human, these cycles are the same way how they function in the bacteria, the same way they function in the humans as well. And almost the same enzymes work in the process. And thus, these constitutive enzymes are present almost in all the cells given in the same concentration at all the time. They are independent of the composition of the medium in which the cells are present. I hope you're clear now with the word constitutive enzymes. So let's proceed with the second type of enzymes. These are inducible enzymes. Now concentrate on the word inducible. Right? Inducible enzymes are the one which are produced in the cell in response to a particular substrate. So these are not always present in the cell. So when a particular substrate comes into the cell, the production of these inducible enzyme is triggered. So you can tell that these inducible enzymes are produced only when the cell needs them. And they are produced only when a particular kind of substrate is present inside the cell. Now, what is the substrate which is triggering the production of these inducible enzymes? So this substrate is called as the inducer. So you can tell a inducer is a substrate which evokes the formation of inducible enzyme. So now you concentrate on the word substrate. So inducer is always a substrate. Substrate of which enzyme? Substrate of inducible enzyme. I hope you're clear with inducible enzyme. Now the third enzyme is repressible enzyme. Now the word repressible is important over here. Repressible enzyme is an enzyme whose synthesis is down-regulated. What do you mean by down-regulated? Means it is stopped. The synthesis of this enzyme is stopped by the presence of its own end product. Repressor enzyme is a one whose synthesis can be stopped by the presence of its own end product. Now, what is this end product called as? This end product is called as a co-repressor, the co-repressor of the repressible enzyme, which is nothing but the end product of this particular enzyme. By this, I think you will be clear with the terms constitutive, inducible, inducer, repressible, and co-repressor. These five terminologies is very important when you are dealing ahead with the mechanism of regulation. If you haven't understood these five terminologies, go back again, see the slide and proceed further, because half of the things will be clear for you when you understand this terminology. Okay, so by this I'll proceed with the next slide. So students, let's deal with the mechanism of regulation of enzymes. So bacteria regulates their enzymes in two broad ways, that is regulation of enzyme activity. Here the enzyme is already present in the system, only its particular activity is regulated, not the synthesis of this enzyme, but the activity of this particular enzyme is inhibited. In this category comes the feedback inhibition or the end product inhibition. Now focus on the words feedback inhibition or the end product inhibition. What do you understand by this? This means that the product of a particular enzyme is inhibiting the activity of this enzyme. So it is basically dealing with the constitutive enzymes. We know that constitutive enzymes are always present in the system at a given concentration. They are vital for the functioning of the cell. They are highly specific in their regulation. Only a particular substance can regulate their activity. And in this, the best example can be given is that tryptophan biosynthesis pathway. That is our TRYPTOPHAN model. Okay, I hope now you're clear with the word regulation of enzyme activity, in which you get feedback inhibition or the end product inhibition, and in that you get the TRYPTOPHAN model. Okay, coming to the second category is regulation of enzyme synthesis. Here the enzyme is synthesized. Okay, the category which is dealing with this is that end product repulsion and enzyme induction. In end product repulsion, focus on this. It is a process where the end product is actually inhibiting the synthesis of its particular enzyme. That is, it is dealing with the reversible enzyme. Okay. And now enzyme induction. Now, what do you mean by enzyme induction? It means that a particular substrate is triggering the enzyme synthesis. So, a substrate is triggering its own enzyme synthesis. So, it is basically dealing with the inducible and repressor enzymes. So the best example here is our famous OPERON model. Now comes the new category in this. It is catabolite repression. It is a form of positive control because it increases the rate of transcription of protein. As of now, you understand so much that it is a positive control because it is increasing the rate of transcription of a protein. We will deal all these topics in detail ahead in the class. Okay? Get familiar with these words and the classification of these. Okay. So students, let's begin with feedback inhibition. Before beginning with this, let's see this diagram. What is this diagram? This diagram is generalized explanation of a biochemical pathway. Here you can see the beginning of a metabolic activity where there are four different enzymes. One, two, three, four are working. In between, the intermediate products are formed, and at the end of a biochemical pathway, you have our end product forming here. So now what is a feedback inhibition? A feedback inhibition occurs when a biochemical product of a pathway blocks an enzyme in the beginning of the pathway. So here, the end product of a biochemical pathway is actually inhibiting the enzyme 1, that is the enzyme in the beginning of the pathway. Okay, when does this occur? This occur when there is a buildup of excess of product in the cell. When there is more product being produced, cell use this mechanism to slow down the production and to conserve the energy, as well as to maintain a state of balance, that is homeostasis within the cell. Now let's look at this enzyme 1. The first enzyme in this pathway is an allosteric enzyme. Now, what is this allosteric enzyme? I will just give you a brief idea about this allosteric enzyme. Allosteric enzyme is a one which has an active site or a catalytic site, a active site and a catalytic site where a substrate can bind in, and a different allosteric site or a regulatory site where an allosteric effector can bind it. Now let's consider this enzyme 1 as an allosteric enzyme. So what's happening when it is an allosteric enzyme? The end product of a biochemical pathway is acting as an allosteric effector, which is binding to the enzyme substrate complex at regulatory site and rendering this entire enzyme complex inactive. Therefore, what happens when enzyme 1 become inactive? The other enzymes, enzyme 2, 3, 4, will remain active. Only the enzyme 1 is inactive because it is inhibited by the end product of this cycle. Though the other enzymes in the pathway are active, they do not see their substrate. The pathway is shut down as long as there is more number of end products present. If the end products are used up or if they get disappeared, the inhibition is removed. There is no allosteric effector. Therefore, the enzyme 1 will regain its activity and the organism can resume the synthesis of the end product. This again, the entire cycle will continue as usual. Okay, the best example for feedback inhibition is tryptophan biosynthesis pathway. Now let's look at this tryptophan biosynthesis pathway. So here is our tryptophan biosynthesis pathway. Let's look at this diagram. This is a very generalized tryptophan biosynthesis pathway diagram occurring in E. coli and showing feedback inhibition. Now let's look at this diagram. There is glutamine and chorismate. These two compounds are combined together in presence of enzyme A to form anthranilate. There are different enzymes here further, like enzyme B, C, D, E, which results in the formation of intermediates B, C, D, and resulting in the formation of an end product called tryptophan. Now, when a cycle is continuously progressing and making huge quantities of tryptophan, there lies a need to control this mechanism. So what happens when the concentration of tryptophan is very high? The tryptophan will act as an allosteric inhibitor, inhibiting the first enzyme in the pathway, that is our enzyme A. Here, this enzyme A is an allosteric enzyme, which is inhibited by the tryptophan, which acts as an allosteric inhibitor. Okay, thus this entire cycle stops. But these enzymes B, C, D, E are active, but they have no substrates so that they can function. So this entire pathway is at halt. Okay, as said, it is a feedback inhibition. The tryptophan is an allosteric effector for the enzyme A, which is an allosteric enzyme. And this is happening when the when the tryptophan concentration is high within the cell. Suppose if the tryptophan concentration is reduced and tryptophan is disappearing or used up, then the tryptophan will disjoin from enzyme A and the pathway will resume. Tryptophan biosynthesis is also regulated by enzyme repression at genetic level. I will be dealing this in next slides when I explain the TRYPTOPHAN model and how this enzyme repression is occurring at genetic level will be explained next. Okay, students, let's look at the tryptophan OPERON model and its control elements. So, what is an OPERON? What do you mean by an OPERON? An operon is a cluster of genes that are controlled by same elements and are transcribed and translated together. So, okay, that means genes which work together are located on the chromosome, and these are transcribed and translated together to effect efficiently. Now here is a structure of tryptophan operon. The genes for the tryptophan biosynthesis in E. coli are organized on the bacterial chromosome in the tryptophan operon. The tryptophan operon consists of the promoter region, an operator region, an attenuator region, and 5 structural genes which are essential for the synthesis of the enzymes in tryptophan biosynthesis. So, let's look at the diagram now. Okay, this is a regulatory gene. It is usually represented as R. This is a promoter region. This is an operator region. This is an attenuator region. And these are the 5 structural genes. That is TRIP A, B, C, D, E. All these genes, especially these 5 genes, they help in the synthesis of the enzymes which are involved in the biosynthesis of the tryptophan. Okay, this is a regulatory genes. In between this promoter, operator, and attenuator regions are nucleotide sequences. Okay, they have a specific function which I'll be letting you know. But as of now, you should know that there is a regulator gene and 5 structural genes, and between you have a operator region, promoter region, and a attenuator region, and they are usually represented by R, O, P, A and TRIP A, B, C, D, E. Okay, this is the basic structure of a TRYPTOPHAN model. So students, let's go in detail with the TRYPTOPHAN model and its control elements, where I'll be explaining the function of the each controlling part in detail. Here, As you know, the regulatory gene, which is located at the first, that is the upstream of the OPERON model, TRYPTOPHAN model, this regulatory gene function is that it encodes a repressor protein. This repressor protein in turns regulates the synthesis of the enzyme, by the five genes. It controls the functions of the five genes, that is TRIP A, B, C, D, and E. So what is the function of this repressor protein? It controls or regulates the functions of the five structural genes. Now, besides the regulatory gene, you will get a promoter region. The promoter region is a nucleotide sequence. It is not a gene, it is a sequence, nucleotide sequence, where RNA polymerase comes and binds. RNA polymerase is an enzyme which initiates the transcription process. Okay, so in promoter region, the RNA polymerase comes and binds. Then you have an operator region. The operator region is also a nucleotide sequence. It is not a gene, it is a sequence where the repressor protein comes and binds. The repressor protein is synthesized by the regulatory gene. And where does it bind? It binds in the operator region. So if the repressor protein binds to the operator region, the RNA polymerase cannot bind to the promoter region. This is a condition. Okay, so when an RNA polymerase cannot bind to the promoter region, the transcription cannot get started. Therefore, the transcription gets inhibited. And none of the enzymes required for the tryptophan biosynthesis pathway are synthesized. Okay, if RNA inhibits translation, the enzymes cannot be synthesized. The structural five genes cannot work. So, okay, as told earlier, TRIP A, B, C, D are the genes which codes for the enzymes involved in the tryptophan biosynthesis. And tryptophan is an end product of this entire pathway. This tryptophan, when it combines with the repressor protein, it makes the repressor protein active. And this repressor protein will go and bind to the operator region. And once it binds to the operator region, the transcription is stopped, and the genes, structural genes cannot express themselves. Thus, the tryptophan is considered as a co-repressor. So, students, let me explain this mechanism with the help of a diagram. So, let's consider a diagram given here below, the below diagram here. Okay, this diagram. So this diagram is, you have a promoter region, operator region, and then you have all the five genes. There is an attenuator region also here, over here, but it is not shown in this diagram, but there is an attenuator region here as well. So in this condition, there is a condition existing now that when the tryptophan is absent. So tryptophan now is considered as a co-repressor. When the tryptophan is absent, it cannot go and bind to the repressor, and the repressor is inactive. Therefore, this repressor is not binding to the operator region. And the RNA polymerase, which binds at the promoter region, is going forward, resulting in the transcription process. And as it goes forward, all these genes are transcribed and result in the synthesis of mRNA. Okay, this condition over here is when the tryptophan is absent. Now let's consider the above picture over here. Now when the tryptophan is present, this tryptophan will go and bind to the repressor protein, and this repressor protein will get active and it goes and binds to the operator region. Earlier here it was inactive, so it was not binding. Now in presence of tryptophan, it is going and binding to the repressor protein, making the repressor protein active, and this repressor protein is now going and binding to the operator region. And now this RNA polymerase cannot bind to the promoter region, and thus the transcription over here is stopping because of the presence of the tryptophan. Okay. So now students, let's consider a normal mechanism of a trypoperon model. Here in this condition, the tryptophan is absent. So what happens when tryptophan is absent? The RNA polymerase enzyme can directly come and bind to the promoter region, and it transcribes. It results in the initiation of the transcription. So these structural genes are transcribed. The transcription results in the synthesis of mRNA strand. These mRNA strand on further translation results in the synthesis of the proteins. What are these proteins? These proteins are nothing but the enzymes which are used for the tryptophan biosynthesis. The condition here was that tryptophan is absent. So where is this trypoperon model located in the E. coli? It is located in this cluster of gene called as an operon on a chromosome inside the E. coli. Okay. I hope students now you are clear with the mechanism of tryptophan operon model, which is an example of a feedback inhibition as well. Okay. So students, let's deal with the enzyme repulsion. It is another concept after feedback inhibition. Okay. In this concept can be explained by taking up again the tryptophan biosynthesis model. Tryptophan biosynthesis model, as said earlier, explained by me, is regulated at genetic level by the process of enzyme repression. Okay. Now, what is this enzyme repression? Enzyme repression is the negative control of the bacterial transcription. Negative control means it is stopping the bacterial transcription. Okay. So when the regulatory protein goes and binds to the promoter region of this tryptophan model, it stops the synthesis of mRNA, which further leads to the decrease in the synthesis of the enzymes. This we have studied earlier just now. So here a condition is taken for an example for enzyme repression. Here a condition is taken where the tryptophan end product is high. In a tryptophan biosynthesis pathway, the end product tryptophan is high. So what does the tryptophan do? The tryptophan will go and bind to the repressor protein. It makes the repressor protein active, and this repressor protein will come and bind to the operator region. And when it binds to the operator region, the RNA polymerase cannot bind to the promoter region. And thus the entire translation is stopped because we need RNA polymerase to bind at the promoter region to initiate the transcription process. Okay. Therefore, thus there is no transcription. And this is an example for the enzyme repulsion. Okay. So the students, the another topic which we studied was an enzyme induction in the synthesis of the enzymes. Okay, the second category of the synthesis of the enzyme, we had talked about the enzyme induction process. Okay, here it is nothing but a process where a substrate or a substance which is similar to the substrate triggers the formation of the enzyme. The enzymes are called as inducible enzymes. Okay, these enzymes are not present in the system. When a substrate comes and triggers its formation, then only they are synthesized. Okay, they are synthesized in response to the substrate, and they are synthesized by the cells only when they are needed. These enzymes, that is inducible enzymes, are usually associated with the degradation of the substrate. The substrate which is evoking their formation is degraded by these enzymes. Okay, the substrates are called as inducers. The entire process where an inducer triggers the formation of the inducible enzymes is called as enzyme induction. The best example for enzyme induction is lactose degradation in E. coli and the OPERON model associated with it. Okay, so this we had discussed in the earlier first slide, and now we are going into a detail with the example where lactose degradation is in E. coli is an example of the enzyme induction. Okay, so students, now let us study the lactose degradation in E. coli. Lactose degradation in E. coli is nothing but a way by which lactose is utilized in an E. coli cell. So here, when the lactose is present outside the cell of an E. coli, or it is present in the medium where E. coli is grown, the E. coli cell triggers the synthesis of these two enzymes called as lactose permease and beta galactosidase. Okay, these two enzymes helps in the utilization of the lactose in E. coli. And when are these two enzymes synthesized? They are synthesized only when the lactose is present in the medium outside the E. coli cell. If lactose is not present, E. coli will not synthesize these two enzymes. And this becomes a perfect example for enzyme induction, where a substrate is triggering the formation of a particular enzyme. And this particular enzyme will degrade the substrate by which it is evoked. Okay, so what is the function of lactose permease? Lactose permease helps in bringing up this sugar inside the E. coli cell, and beta galactosidase helps in the breaking up of lactose into galactose and glucose, where the further the E. coli cell will convert this glucose into galactose and use it in other biochemical pathways. Now, why these two diagrams are taken in the consideration? It is because to explain this point, that is, the genes which are responsible for the lactose permease and beta galactosidase are located within the OPERON model. And this OPERON model helps in the lactose utilization pathway in the E. coli. I hope you are able to connect this and understand. Okay. Thus, this lactose degradation becomes an important example of enzyme induction, where the lactose triggers the formation of lactose permease and beta galactosidase, which further degrades the lactose itself. Thus, the enzyme induction. So, students, now let us study the structure of OPERON model. The OPERON model consists of a regulatory gene, which is called as LAC-I. This LAC-I is shown here in the diagram. This is the regulatory gene. This regulatory gene encodes repressor protein, that is repressor RNA, and this repressor mRNA is further translated into a repressor protein. Okay, this repressor protein regulates the genes present on the OPERON model. So Here in this diagram, you have to understand that this part is called as a regulated gene called as LAC-I. This is the promoter site. This is a nucleotide sequence to which RNA polymerase comes and bind. It is similar to the TRYPTOPHAN model, where in the promoter region, the RNA polymerase comes and binds. And this is the operator region. Here, this repressor active in its active states comes and binds at the operator region. Okay, the difference here between the TRYPTOPHAN and LACTOPHAN model is this: the promoter region here is divided into two parts. You can see here, this is also a promoter region, this is also promoter region. In between, you have a regulatory gene. Okay, so this is called as a upstream and this is downstream. The upstream of the promoter site is called as a cap site, and the downstream here is called as the RNA polymerase binding site. So the RNA polymerase comes and binds here, just like the TRYPTOPHAN model. And here you have three structural genes, that is LAC-Z, LAC-Y, LAC-A. So LAC-Z is used for the synthesis of beta-galactosidase enzyme, and LAC-Y gene is used for the lactose permease enzyme, and LAC-A genes is used for the transacetylase enzyme. Okay, so these three genes synthesize an mRNA, and this mRNA on translation will result in these three enzymes. Okay. So students, let's understand the mechanism of LACTOPHAN model in simple terms. Okay. Till now, what we have understood is that we need lactose present in a medium so that the E. coli can trigger the synthesis of the two enzymes called as lactose permease and beta-galactosidase. So when the lactose is not present, the E. coli is not synthesizing these two enzymes. And how is it possible? Let's look at this OPERON model above this figure. Here, in the absence of the inducer, that is, in the absence of the lactose, the regulatory gene is synthesizing the repressor protein, is encoding the repressor protein. This repressor protein straight away is active, and it straight away goes and binds to the operator region. And when this goes and binds to the operator region, the RNA polymerase cannot bind at this promoter region. We need RNA polymerase to initiate the transcription process. But when the RNA is not binding here, the transcription process is not started. Thus, the enzymes are not synthesized. Therefore, in absence of lactose, the enzymes are not synthesized. But when the but when the lactose is present, the substrate lactose is present, it goes and binds to this repressor protein, which is synthesized by the regulatory gene, and make this repressor protein inactive. Once this repressor protein is inactive, it cannot go and bind to the promoter region. Therefore, the RNA polymerase directly comes and binds at the promoter region, and the transcription takes place. And these three structural genes are transcribed. LAC mRNA is generated, and this LAC mRNA on further translation results in the formation of three proteins. Those are nothing but our enzymes: beta-galactosidase, lactose permease, and transacetylase. Okay, and these two enzymes, beta-galactosidase and lactose permease, are helpful in the lactose utilization in the E. coli. And this is possible only when the lactose is present in the medium outside the cell. Okay, I hope the explanation is clear. So students, now let us deal with the last topic in the mechanism of enzyme regulation. This is called as catabolic repression. It is also called as glucose effect because it was first discovered during the study of regulation of OPERON model. Okay, now let us consider the first graph. Here, the E. coli is grown in two separate mediums. One medium is containing the glucose, the other medium is containing the lactose. So you can see when a graph is plotted with number of cells versus time, you can see there is an exponential increase in the number of cells with the time. And similarly, there is an increase here in the lactose medium as well. But when compared to the number of cells in glucose medium is high when compared to the cells growth in the lactose medium. This first graph shows that glucose is primarily the first choice of E. coli. Okay, whenever glucose is there, it is their first choice, and lactose is the second choice. So now in the second graph, let's see where an E. coli is grown in a medium containing both glucose and galactose. We have understood by the first graph that glucose is there primarily the first choice and lactose is the second choice. Now what will happen? Here, there is a result of a diuretic curve. Okay, this curve consists of the first phase and a lag phase and a second phase. This first phase is called as a glucose utilization phase. In this phase, the entire glucose is utilized and the cells are growing at an exponential rate. A time comes where all the glucose is consumed. Okay, and then after some time, a lag time, there is no growth, and then you can see a second phase where the lactose is utilized is there, and again there is an exponential growth of the cells. Now, what do you understand by this? Here, we understand until and unless the entire glucose is utilized, there is no utilization of lactose by the E. coli. And how is it happening? Because when lactose is present in the medium, we have studied in OPERON model that when lactose is present in a medium, it triggers the formation of the enzymes which use this lactose, right? But here, the cell is not utilizing the lactose. Why is it? This is because the glucose is inhibiting the OPERON model. Until and unless the glucose is present, the cells cannot utilize the lactose. Therefore, once the entire glucose is utilized, then in this lag time, where there is no glucose left, the cells are synthesizing the enzymes required for the lactose utilization in E. coli. The two enzymes which we know is lactose primase and beta galactosidase are synthesized in this lag time. And then here you can see the exponential rise of the number of cells on utilization of lactose. Thus, the catabolic repression is seen by the glucose on the lactoperon model. Thus, catabolic repression. So, students, we are done with the mechanism of enzyme regulation. We have studied feedback inhibition, enzyme induction, catabolic repression, the OPERON model, the tryptophan model, and allosteric enzymes. Okay, and from this, what are the questions which we can expect? We can expect the question, the mechanism of enzyme regulation in bacteria. Then the mechanism of feedback inhibition, the process of enzyme repression, the TRYPTOPHAN model and its control elements, explain the enzyme regulation by enzyme induction, the OPERON model and its control elements, the given account of catabolic repression and allosteric enzymes. So all these questions are covered in this particular topic. I hope you were able to understand. This topic is one of the trickiest, as I said earlier, but I have tried to give you an easy explanation. Hope it will help you all. Okay. Thank you for listening to my class. Thank you.